Good morning, distinguished guests, ladies and gentlemen. It is my great honor to welcome you all to this highly anticipated awareness seminar. Before we begin with the introductions of our special guests of honor, allow me to provide a brief overview of the initiative that brings us together today. Install Africa. Install Africa, the innovation, science and technology for action and learning in Africa, is the result of collaborative efforts initiated in October 2023. This initiative leverages the Drosophila melanogaster fly model and state-of-the-art infrastructure to enhance fundamental research capacity in Rwanda. Rwanda has made remarkable strides in advancing its scientific capabilities, recognizing the pivotal role of research in sustainable development and public health. Install Africa was born from a collaboration between the Foresight Institute of Research and Translation, Rwanda, FIRAT Rwanda, and its technical partner, DROS Africa UK, in conjunction with key national institutions such as the National Council for Science and Technology, Rwanda Biomedical Center, the National Industrial Research and Development Agency, the University of Rwanda, and the Rwanda Food and Drug Authority. This collaboration embarks on a 5-year comprehensive capacity development plan aimed at cultivating the next generation of scientists and fostering research excellence across medical, applied, and fundamental sciences. From healthcare innovation to agricultural production, this partnership envisions driving progress in public health and sustainable development across Rwanda and beyond. It is against this background that we convene today, united in our shared commitment to advancing science, technology, and public health for the benefit of our communities. With that foundation laid, I am now delighted to introduce our special guests of honor, whose presence today underscores the importance of this endeavor. Today, we are privileged to have with us an exceptional panel of leaders, experts, and visionaries who have made remarkable contributions in their respective fields. Allow me to introduce our esteemed special guests of honor. First, we are honored to welcome Professor Abraham Mutasa, the Principal of the College of Medicine and Health Sciences, University of Rwanda. Professor Mutasa's leadership in medical education and research has been a beacon of progress in the healthcare sector. Next, we have Dr. Eugene Mutamura, the Executive Secretary of the National Council for Science and Technology. Dr. Mutamura has been at the forefront of advancing science and technology policies that shape Rwanda's development. Joining us as well is Professor Claude Muvunyi, the Director General of Rwanda Biomedical Center. His dedication to strengthening public health systems has profoundly impacted the nation. We are also privileged to have Dr. Sekomo, the Director General of National Industrial Research and Development. Dr. Sekomo's work in fostering industrial innovation and development is truly commendable. Another distinguished guest is Professor Emile Bienvenu, the Director General of the Rwanda Food and Drug Agency. Professor Bienvenu's contributions to ensuring the safety and quality of food and drugs in Rwanda are invaluable. We also have Dr. Dieudonné Mutangana, the head of the Department of Biological Science at CST University of Rwanda. Dr. Mutangana's expertise in biological sciences has been instrumental in academic and research excellence. Finally, we are delighted to introduce Professor Janine Kondo, the CEO of the Center for Impact, Innovation, and Capacity Building for Health Information Systems and Nutrition, CIICHIN. Professor Kondo's leadership in health innovation and nutrition has made a lasting impact locally and globally. Please join me in giving a warm round of applause to our special guests of honor, whose presence graces this important seminar. Next, we will move on to narrating the profile of our invited guest speaker, Professor Leon Mutesa. Professor Leon Mutesa, MD, PhD, is a leading authority in the field of human genetics, renowned for his transformative impact on research, education, and healthcare across East Africa and beyond. As a professor of human genetics and the founding director of the Center for Human Genetics at the University of Rwanda, he has led groundbreaking initiatives that have revolutionized genetic research and established vital infrastructure for medical genetics in the region. His unwavering dedication to advancing knowledge and improving access to genetic services has solidified his reputation as a cornerstone of progress in this critical field. Professor Mutesa's journey began at the University of Rwanda, where he earned his medical degree in 2003. He later pursued a PhD in human genetics at the University of Liège, Belgium in 2009, followed by a postdoctoral fellowship at the same institution in 2011. His advanced training and relentless pursuit of excellence have propelled his expertise across diverse domains, including molecular genetics, cytogenetics, epigenomics, precision medicine, forensic science, and public health genetics. Throughout his illustrious career, Professor Mutesa has demonstrated exceptional leadership in academia, clinical practice, and health sector management. As Director of Clinical Laboratory at Kigali University Teaching Hospital, Rwanda's largest referral hospital, he significantly enhanced diagnostic service quality. Later, as Director General of the Medical Research Center at the Rwanda Biomedical Center, he played a pivotal role in integrating research into national health strategies. Under his leadership, the establishment of the Rwanda Health Research Commission streamlined research processes and ensured adherence to ethical standards. On the international stage, Professor Mutesa has spearheaded collaborative research initiatives, serving as principal investigator on prestigious NIH-funded projects, including the H3 Africa program, which examines the transgenerational epigenomics of trauma in Rwandan genocide survivors. His leadership in the NIH NCIU 54 project addresses the prevention and treatment of HIV/HPV-related cancers in Rwanda, with a focus on cervical cancer in HIV-positive populations. His expertise has been sought by global organizations such as UNICEF, UNAIDS, Africa CDC, and WHO, further solidifying his role as a global authority in genetics and health. Professor Mutesa's research output is nothing short of extraordinary, with over 200 peer-reviewed articles published in leading scientific journals. Among his most notable contributions is a groundbreaking study on cost-effective SARS-CoV-2 diagnostics, published in Nature, which detailed an innovative pooling strategy to reduce testing costs during the COVID-19 pandemic. His research on trauma epigenomics, precision medicine, and infectious diseases continues to shape global scientific understanding and influence public health policies. In addition to his research, Professor Mutesa is deeply committed to capacity building. He has mentored over 90 postgraduate students, fostering a new generation of scientists prepared to address Africa's health challenges. Through his leadership of various consortia, he has strengthened research capacity across the continent, ensuring Africa remains at the forefront of scientific innovation. His current research focuses on the epigenomic effects of trauma, advancing cancer genomics in high-risk populations, and integrating precision medicine into healthcare systems. His work on newborn screening programs and scalable diagnostic strategies reflects a vision for healthcare that is both innovative and accessible. His dedication to forensic science and cytogenetics further demonstrates his multifaceted approach to understanding human genetics. Professor Mutesa's achievements have been recognized through numerous awards and global appointments. Among his prestigious roles, he serves on the Board of Trustees of the International Vaccine Institute. His career exemplifies visionary leadership and scientific rigor, driving meaningful change in genetics and public health. As he continues to advance research and health initiatives, Professor Mutesa stands as a beacon of progress, championing the integration of cutting-edge science into sustainable healthcare solutions. We are honored to have him with us today to share his insights and vision for the future of health and science. Professor Mutesa, the floor is yours.
Good morning, good afternoon, depending on where you are. And I'm really delighted to be here, and thanks very much for the nice introduction. I just first of all want to thank the organizers of this webinar, recurrent webinar, which just allow the Rwandan scientists to share the experience, but also the advances in the different research perspective in Rwanda. So today I'm just honored to share the actually it's an experience that we began during the recent outbreak of Marburg. So today I have a topic on genomic characterization of the Marburg virus, which was one of the good example to show the international community how Rwanda has been just able to overcome this outbreak with a rapid response. I just want to start by saying that this outbreak, as you have been seeing in different newspaper and medias, was just linked to zoonotic event, and then we came to be able just to show this linkage between the transmission between the human and the animals, which was a very good example for when you compare to previous outbreak that happened in the world. So there's a lot we can say about the Marburg outbreak in Rwanda, but today I choose just to talk about the genomic characterization, which allowed the country to uncover the transmission dynamics of this virus and following this single zoonotic spillover event. As you may know, the Marburg virus has been identified for the first time in Germany in 1967, and this virus is a member of Phylogenetic family, where you see other type of viruses like Ebola. And then during that time, 1967, it was just identified from an outbreak that was declared from a lab activity where there have been a contamination of lab workers from origin of from an African green monkey, which was just imported in that country at that time for pharmaceutical use. And then that plague caused a lot of fatality cases at that time. So, and then it was the first time to see the disease with severe hemorrhagic fever. And then it was named the Marburg virus because it was identified in that town of Germany, in Marburg. And then later on, other outbreaks happened all over the world. And then the case fatality has been estimated between 23 to 88%. You can see this is very high number. Also depending on the outbreak and the healthcare access and infrastructure to contain the outbreak. And when you zoom in the genomic structure of this Marburg, you can see on this structure in this picture different genes, VP 35, 40, 24, and so on. Those are the structure that characterize the genome of this virus. But interestingly, there are two genes that I want just to focus on, the VP40 and the GP. Those two genes that are like the heart of the virus. I'll show you later why. These are even the target for vaccine design. And in total, the virus has 7 genes which are subject to change, generating different variants, different mutations. That's how the normal cycle of the virus operates during the process of replication. And then if you go one by one on these genes, there are some attributes and the functions. But why I mentioned especially VP40, that means viral protein, the GP, which is glycoprotein. Those are the essential genes that we see in this genome, which are target not only for vaccine design, but also other drug design, but also when we design the diagnostic tools for testing. So I will not just go into details, but you see the reference if you want to have more details about each gene of these 7 genes of this virus. And then this virus, as I mentioned, there have been previous outbreak. Rwanda also registered the recent outbreak, but you see already over the world different outbreaks that happened. In the red zone here, especially in the African continent, those are the outbreaks that have been declared as an original strain from the specific country where the outbreak has been just declared. That means there has not been any information about imported cases. It has just been declared in that country. So while when you go to the yellow countries like Russia in 2008, Germany as well, 1967, I mentioned this, South Africa. Those are the outbreak that has been imported. Those were like imported cases. And then the green zone here in Russia, sorry, here I mentioned the yellow, that was USA. In Russia, that the outbreak which happened in since 1988 up to 1995, which was an error during the laboratory work. It was just a laboratory exposure, and then this resulted in the outbreak for the whole country. It was also a disaster at that time. And this outbreak, this disease, we know it's just transmitted. It can be transmitted from the animals. I will show you clearly in the next slide. But it has different type of strains from different reservoir. The bats species, those are the common reservoir for this disease. But the most common reservoir bats is Rosetta aegypticus, which is a fruit bat species, and you can see different periods of the outbreak that I mentioned earlier, from 1999 up to 2018, in different countries like Gabon, Kenya, Sierra Leone, Uganda. Those have been especially caused by the reservoir from the fruit bat, this Rosetta aegypticus. So we see what happened to Rwanda as well later. And regarding the transmission and the spread of the Marburg virus, there can be a direct contamination between bats, either through the sexual transmission or by biting. Then the Rosetta aegypticus also can have the direct contamination between this bat and the humans through different body fluid like saliva, urine, and also other contaminated items like fruits, because this category of bats, they really like fruits, and most of the outbreaks have been shown that the people have been contaminated through that mode of transmission. And then can also be human to human transmission. This is a direct contact of body fluids like semen, slight possibility also via air respiration. But again, human to human, but also other primates, non-human primates can also contaminate between them, also between but also between humans, between human and the non-primates. Those modes of transmission have been reported previously. What about the clinical symptoms? Actually, this outbreak, when there's an outbreak, when there's the contamination, there's a period ranging between 5 to 21 days where you have an incubation period, and the infected individual cannot just show the symptoms. However, sometimes when the incubation period is over, they start to have, during the first 1 or 5 days, general symptoms characterized by high fever, headaches, chills, malaise, fatigue. And those are really the similar symptoms with other tropical disease like malaria. So in this period of the disease, sometimes there is a confusion or also problem just to miss the diagnostic by confusion with the other tropical disease. And then there will be evolution after the 5 days, where you see during the first week and the second week, up to 13 days, 14 days, the hemorrhagic symptoms being dominating. And then in these hemorrhagic symptoms, I'll show you on pathophysiological level what's happened. The different organs will be affected, and then there will be hemorrhage characterizing this disease. And then if nothing is done, this will lead to common complications during the second and third week with general symptoms and a common complication where there will be multi-organs failure, like kidney, liver, spleen failure just happening. This is really the most cause of the death for the affected patients. But most of those patients who have been recovered, they don't reach this stage, late stage, which is very fatal for the affected persons. And then I briefly mentioned how the mode of transmission operates. On this slide, you can see the virus entry and the viral transmission, dissemination, and the cellular tropism. And the yellow box color, the figure here on top, this shows the viral entry mechanism, while the red color here shows the viral dissemination pathway. For this yellow box, the virus entry will happen by skin lesions or abrasion. And then this will allow the virus you see here in green to enter through the screen, and also to have cell depletion by destruction of the spleen and the lymph nodes. And then the virus will enter the host and spread the flu throughout the lymphatic and the vascular systems. You see here in the light brown color, indicating the damaged cellular organelles. And then the Marburg virus will cause the necrosis in many organs. You can see here necrosis in many organs, including liver, spleen, gastrointestinal tract, and endocardium. And then the hemorrhagic fever and also the hemorrhagic pattern will happen later after just this dissemination pathway. And on this slide, you can see different organs which are going to be affected. Actually, the Marburg virus primarily we targeted the dendritic cells, monocytes, parenchyma cells at the liver function, and adrenocortical cells, as well as several lymphoid tissues. For the dendritic cells, the infection will lead to poor stimulation or stimulating condition of T cell lymphocyte, which will cause the lymphocyte apoptic condition. Due to this, then the body's immunity is suppressed, and the cytokines and the chemokines number is increased, which will lead to a shock and as well as a multi-organ damaging happening. And then secondary, I mentioned the macrophage and the monocyte here. The infection will lead to uncontrolled cytokine and the chemokines activation. And then they will continue the damaging of the T cell, lymphocytes, and endothelial cells. The endothelial cell infection will then cause increase of blood vessels permeability and DIC, the disseminated intravascular coagulopathy will happen, while both occurrences will lead to the hemorrhage. See, this is you can see on the bottom of the slide always the hemorrhage. That's where we call it hemorrhagic fever. So what happened actually in this case, the blood vessels permeability, which will increase, that will allow the blood just to be leading to this hemorrhagic pattern. And then the parenchymal cell infection will occur in the liver, and then this decrease the coagulation factors, and this will occur. The occurrence of this can also cause the hemorrhage later on. Adrenocortical cells and adrenal gland infection occur by this virus, which will lead to the disorders in the metabolism and dysregulated blood pressure, and the hemorrhage will also occur on a later stage. And finally, on the right corner of this figure, you see the lymph nodes and the spleen being affected. Then the infection of these lymphoid tissues and of lymphatic system, especially the lymph nodes and the spleen in this case, will lead to the tissue necrosis and the malfunctional adaptive immunity. And this will lead to a shock and the lymphoid organ damage, which will occur in the large stage. So as a take-home message on this slide, very complicated, you see the multi-organ failure, and then all this organ will lead to hemorrhage. That's why we call it hemorrhagic fever disease. Now, so that was just a background about the disease. Then what do you do in case there is the declaration of the outbreak? First of all, you think about the personal protective equipment, which will allow the health professionals just to tackle and to contain the outbreak. What you have to know, actually the PPE alone is not enough. In this case, safe patient care during the Marburg virus outbreak will require also other standard precautions, including hand hygiene, environmental cleaning, screening, and isolation. And the PPEs, the use of PPEs in the standard precaution during the virus, the Marburg virus outbreak is also common to other outbreaks. So yeah, just to give you an information, the precaution need to be activated in this context. And some of the good examples of the items, but the most appropriate here is this cover our body, where you just avoid any contamination in the area where you are just managing or treating the patients. And of course, for the Marburg virus screening, can be, if the screening can be a non-contact activity. For example, if the distance can be maintained, then there's no need just to use the PPE, but in a case this can't be maintained, then the PPEs are recommended. This is just the first thing to do, first activity to do when you want just to manage the outbreak. So just this picture also shows you different PPEs and cleaning activity, environmental activity, but also how you need just to wear this one. Number two, especially in this context of testing, because my talk is just focusing on this. Maybe next time we'll be talking about the management of patients or also vaccine of the patient. So in this context, lab testing is a key. This is the heart of the management of the outbreak. If you don't have a very high standard laboratory, we will not be able just to get the reliable results and also to manage which can inform the policy makers on the case management and even the surveillance team, just how we manage the disease. Normally, you need to have to be able just to contain this kind of outbreak. You need level 4 B cell, level 4 lab, but in a context setting, clinical setting environment like Rwanda, where we don't have level 4, at least you can just design a lab quickly to contain this. This is just an overview of how we designed quickly our labs. You can have an idea how the lab has to be designed, even how it looks like for the level 4. This on the level 3 in our context. You have the entry and the exit, which is in the common here, common area, and then changing room where the health professionals, lab technician will put their PPEs, and then you go to testing floor 1. This is a dedicated room for the contamination and the inactivation of the virus. Remember, we are dealing with a very dangerous virus. So the safety procedures are key in this context. And then you leave testing room floor 1 to floor 2 for RNA extraction, and then you continue to floor 3 for master PCR, master mix. And if you also want to do the preparation for sequencing, this is where it happens, and then you finish by the last room for PCR amplification or sequencing, and then you exit. So this has to be like a single way, not just double way, to avoid contamination, as I mentioned in this context. This is how our lab had been designed just to contain the outbreak. For diagnostic and detection, remember this, at early stage, the symptoms are similar with other diseases like malaria, typhoid, dengue. Just need to be careful. And then you needed quickly to request for RT-PCR to see if there is a positive case or a positive sample, and although you can do ELISA to see if there is a detection of antigens or antibody on this point of care testing. And then remember why I insisted on the genes. So you can see the VP40 here. This gene, this is really the heart of the disease. If we have a kit in the lab, RT-PCR kit detecting this, the presence of this gene, then this tool or this kit is having a higher specificity and sensitivity, which is a very key just to focus on this. So on this picture on the left side, you see different genes which are being also targeted for different diagnostic perspective, but also even for the Ebola virus, you see, which is a bit similar. The gene is a bit similar to the Marburg virus genome, but the kits have been just designed on targeting the genes that you see. And then what happened really in this country, we did characterization, genomic characterization of this virus from the samples that have been identified. during the genomic sequencing. So this picture shows you the outbreak response. The first laboratory, this is really a timeline response, how all activities and the track have been just activated. The first laboratory confirmation happened end of September on 25th. And then National Public Health Emergency was declared on 27th, and the community door-to-door started on 1st October. And then our genomic sequencing started just during that week on 7th October. You see now the response has been very rapid. In addition, you can see on the peak, the blue peak, that's the peak of cases, the identified positive cases. And the red one, those are the deaths. And then the green, those are the recovered curves. So, but you see, we registered a high level of cases during the first 2-3 weeks of the outbreak. And then when we have reactivated the responses, we started just to see the decrease of the cases, which was really good. And that's how the case fatality rate was really at a low level, 23%. You see only 15 deaths out of 66 confirmed cases. This was really the first time in the world having this type of Marburg outbreak, being contained, also having a low case fatality. Other, if you just you see different previous outbreak, the case fatality has been significant, over 88%. This is a remarkable work done here. And then coming back into our lab, then for diagnostic, we did, of course, TPCR testing. This is the testing flow. And then sequencing, doing metagenomics of amplicons and the metatranscriptomics. This actually is happening now. I will just show you on our last slide. And then behind this, addressing bioinformatic analysis quickly. And now, as you know, this is a linked zoonotic link spillover event. And then One Health component came in. Actually, we are busy working on this One Health component. Now, what did we do? For sequencing, we use different platforms, Illumina platform and Oxford Non-Pol Technology, ONT. So this platform, they were complementary just to provide a very comprehensive understanding of genome coverage. So for ONT, we have a long read and then short read from the Illumina. We did select the patient from the two clusters. Actually, there were two big health facilities where the outbreak was contained. That's where we recruited our patient. And then we sequenced the 62 positive and negative samples. And the 28 samples had a very high genomic coverage, more than 80%, which is good just to proceed for bioinformatic studies behind. For the long read and the short read, the interest of doing this is just to increase the high coverage of genomic testing, genomic sequencing. For example, the short read, there'll be missing sequences data, which will be covered by the high read, just to cut the story short. Both technology have been just used on our platforms. And the quality genomic coverage across the new infections were also cross-checked here. What you see on this figure, you see different CT values from different patients, different sequences. So you can see from 12, ranging from 12.5 up to 30. So from the fresh CT values to odd CT values were ranging within genomic coverage we were really targeting. In general, the good quality of this genomic coverage we really observed in all these samples that we sequenced, and over 80%, which was really a very good record in this context. And as a result, most crosses, you can see on our phylogenetic study, we found that the most closest strand identified in our context was related to the sequence from the 2014 Uganda outbreak. Just here on figure A, you see the maximum likelihood of this on this figure of phylogenetics, just showing a crosses between Uganda and the Rwanda outbreak. And interestingly, on figure B, the similarity here was just by the fact that the 2014 outbreak in Uganda, the cases were just linked to zoonotic spillover event from mining cave, where the patient, most of the patients were identified. You can see on the picture here on the map, Piton Cave and the Kitaka mining cave. Those are in Banda district in Uganda, where the cases have been identified. And very interestingly, I will show you to the next slide. Our index case was also related to this event. And surprisingly, during our sequencing, we identified 3 synonymous mutations in non-coding regions in the 2 genes, GP, glycoprotein, and the VP24, viral protein 24 genes. And on figure A, you can have the essence of how these sequences have been just showing different mutation on figure B from different patients. And even on figure B, you can see the onset of the symptoms, the date, and also the date of data collection and the sequencing, which is very interesting. But these 3 mutations were just located in non-coding regions. And we also went far to have just what we call time to most recent common ancestor. Again, on figure A here, you can have the root-to-root comparison of the closest human to zoonotic sequences from Uganda, which are also the same from figure B, related to this closest event between the sequences that we identified in Rwanda. Figure B, you can see the overlapping. And this clearly just draw the roots for this most common recent common ancestor with the 2 outbreaks. And even we try just to have the information for the source of the entry. We could see the similarity amongst the samples that we sequenced, which indicate the source of the single source of entry, which shows a conserved genome from even 2014 up to 2024. You can see that this trend has been conserved over time. That means that the virus did not really undergo several mutations, which is good. But on one hand, it's very good, but on the other hand, that means we need just to increase the One Health activity and surveillance. Then One Health. So here I told you that the transmission between human to human can happen, which happened, but the first case has to be identified. We identified our first case, which was linked to One Health. Actually, that was a mining, someone working in this mining cave on figure A with more than 20 years exposure in this mining cave, which is not far from the capital city of Kigali, where the cases have been just identified, like 7 to 10 kilometers. So you see, it's not really far. And then we went deeper to understand the story, and then we could just trace that case, that the index case was a relative of that man who was working in this mining cave, who has been exposed previously. Before that, his relative just got sick and then unfortunately passed away from the hospital where the cases have been declared. But fortunately for this case of this person working here in this mining cave, he recovered, but we could just test the Marburg virus antibodies, which were positive. And that was really our scientific evidence of the linkage between the human animal to human transmission and linked to this mining cave activities. So here what we are showing is the ongoing activity where we do trapping for fruit bats, the Egypticus, Rhezotus egypticus bats. And on figure C, you see the sampling from these bats. And as of now, we have tested more than 600 samples from these bats. It's a really very large colony. When you go in this cave, more than 4 million of species living there and carrying different mutation, different viruses, types of viruses, not only Marburg, but also other types of viruses like coronaviruses. So this is ongoing. Up to now, all of these samples have been negative, but we are just keeping sequencing, sampling, and also sequencing for this case. And we're happy just to share with the international scientific community this story that I'm telling you. So this is a preprint paper that you can access, and even our sequence are published in the NCBI, the gene bank. You have the accession number here, but you can have the full paper with full information from this preprint on Medrix. And the paper is in the process of being published by a journal. So that's really a very good achievement. Again, I mentioned for this One Health, we are busy doing the metatranscriptomics, trying to understand the type of the Marburg from these fruit bats. And also with the genomic, addressing the genomic diversity, with other targets to have analysis for host population structure and expression of profile from the host component. I can't finish without just talking about a little bit about the treatment. There are different treatment options, but the supportive care from ICU, this is important, where you see some experimental treatment also have been used even in our context. Monoclonal antibodies, these are very useful. It's a little very, I can say, first line treatment for positive cases. And antiviral drugs like remdesivir, favipiravir, those have been also used, investigated. They are still, even though they are still under clinical trials, but they provide really a very good outcome for patients, especially patients who recovered. Actually, most of those patients with our rapid response from the case management, they recovered due to these drugs. But the vaccine development, this is very interesting. And before I talk about the vaccine development, for those who recover, this is how the antibodies just looks like. So you can see the IgG will last for longer, while the IgM and the other will just decrease over time. So actually, the good thing for this, we can use this the plasma from this recovered patient to design other drugs for patients. This is a very good outcome, very good information to consider. For the different therapeutics and the vaccine options trials for non-primates, there's some vaccine trials. I just wanted to show you a list of this, but the list is not exhaustive. But what is interesting is just to see in non-primates, human primates, like macaques, rhesus, and other types of non-human primates, the survival rate on the last column. So you see 100%, 80%. So this is very promising. And even for other species in this category. So you see always there's a survival rate close to 100% during these preclinical studies. And then for in humans, there are some clinical trials, ongoing clinical trials, especially for therapeutics, where you see some preclinical phases completed and others ongoing with different molecules. Even Lemensivir that I mentioned, you can see the phase when we registered our outbreak here. So these are the updated information, but you can have full information from this paper published this year. And for vaccine trials, they also have different vaccine trial in pipeline. The most important is this one, phase 1. Now, the first one, which is now in phase 2 in Rwanda, this is CAD3. It's just adenovirus vaccine, which was tested from the chimpanzee. And we received this vaccine during the second week of the October, and we started just enrolling patients for phase 2. And then the preliminary results are really promising. So it's an ongoing clinical trial which will really change the paradigm on how we treat this and also we prevent this Marburg to happen. So also there are other different therapeutic phases that I mentioned here. Just the list is not exhaustive, as I mentioned. All right, I think I want to finish here by acknowledging different efforts from different partners, especially the Rwanda Joint Task Force led by Minister of Health, RBC, the collaboration with University of Rwanda, RDB, and the other different development partners, UN agencies, WHO, UNICEF, and others. But I also want to acknowledge the contribution from African Center of Excellence for Genomic Infectious Disease, led by Professor Christian Happy, who contributed a lot, especially for supporting the bioinformatic analysis of this, and also the contribution from Africa CDC, led by Dr. Sophonias Tesema, who also brought that technical and scientific, even financial support to contain this outbreak in this country. And thank you very much for your understanding and listening. Now, if there are any question, I'm happy just to answer different questions. So I can see from the chat box. Okay, when I scroll from the chat box, I don't see any questions. So I'll be happy just to answer any question if you have. Otherwise, you can reach me out through the different platforms. I can just share with you my email here. Let me put in capital letter just to see. Yeah. Maybe you can type your question here in chat box. I'll be happy just to answer any question you have. I don't think so. There's a possibility you can speak.
Hello, Prof. Can you hear me, sir?
Oh, okay. I can hear you now. So,
Yeah. Thank you.
yeah,
Thank you very much for the wonderful presentation you have given to us. I can assure you that this is very much inspiring and thought-provoking. You have given us a lot in a very few minutes, and I can tell you that you have given us too much of information to process at the same time. However, for those
oh,
people that probably joined late, so I'll just run through what the professor has presented to us in very few minutes so that I don't, do another lecture over lecture. So the prof talked about the history. It took us the memory lane how Marburg started in Germany as a result of the importation of some African green monkeys for pharmaceutical research projects. And he did talk about the fact that the Marburg virus is a single-stranded negative sense RNA virus with 7 genes. And then he talked about the outbreak, the original source, the imported, and the lab, sources that occur throughout the world in the past. So he also talked about the fact that the Marburg virus is being transmitted by food baths, and also it can also spread across by sexual intercourse, by mixing of body fluids. So he did talk about the symptoms as well. The fact that the symptoms are very much similar to some other tropical diseases like malaria, dengue fever, and all that. So, and the symptoms always come with headache, chills, malaise, fatigue, abdominal pain, etc. So he did talk about the several stages, the multi and culminating into multi-organ dysfunction, and consequently, the leading to increased permeability as a result of damage to the immune system, immune cells like the dendritic cells and the T cells, and all other mechanism at which this virus is impeding immune system. So all this culminates into increased permeability, hence hemorrhagic fever, as we see. So he did talk about the take notes. He told us that the multi-organ failure leads to the permeability, and hence the hemorrhagic fever that we see, or disease that we see in patients. We did talk about the management, the PPE, the good hygiene, the hand hygiene, the environmental cleaning, and all that. I don't want to go into details. Professor, you know, proceeded by talking about the molecular lab testing flow, showing us the from flow 1 to flow 4, how the lab was set up, because to do this work, we actually need about level 4 biosafety cabinet. So it did show us how the lab was structured from flow 1 to flow 4. We talked about the diagnosis. That one needs to be very careful because this virus is very contagious. So he talked about the molecular diagnosis using RT-PCR, and then the search for the L gene, which is the housekeeping gene for the Marburg virus. So he did talk about the genomic sequencing and the fatality demography, with Rwanda having 23%, which is very remarkable. I think Rwanda is leading the pace now, and
yeah.
I think Rwanda will hold the haze for a long time with all the development happening in Rwanda. So he did talk about the flow from the PCR to the sequencing to the bioinformatics and culminating into One Health, because this
Yes.
is a zoonotic infection. So Prof did talk about the closest resemblance, the result of what was obtained in Rwanda having a closest resemblance with 2014 outbreak in Uganda. So, and then
Yeah.
it was amazing that there was 3 synonymous mutation in GP and the VP24 genes, So not to waste our time or not to talk too much, I can I don't have to do another lecture. Prof did closed with the fact that there are a lot of ongoing activities as regarding clinical trials for vaccine development. Thank you very much, Professor Leon Mutesa. And I believe that I am telling you that we have really gained a lot from your presentation. Thank you very much once
Thank
again. So let me
thank you
see if
very
there
much.
are questions, sir. Yes, sir,
Yeah.
you're welcome,
Thank you
sir.
very much. My
So
pleasure. My pleasure
yes,
to answer any question here. Mm.
yes.
Yeah. In any case, this presentation is available. It's publicly available. You can share with each and everyone who wants just to have
Okay,
access.
because I can see that someone is requesting for the presentation already. So
Yes.
someone
Yes.
is
Yes.
saying
That,
here,
that's
yes, thank you, sir. Someone is asking here, sir. Thank you, Prof. I wanted to ask if the genome you have sequenced have a plan to study the factors associated with virulence of the virus at genomic level, comparing isolate from patients.
good question. Actually, yeah, the three. That's a very interesting question. If you have seen from my presentation, there are three synonymous, non-synonymous SNP that we identified in non-coding regions, which could be maybe located in a splicing site. For those who are in the field, there's a gene and then introns. So the gene, this is the coding region, and then introns in non-coding regions. Just on the starting of the non-coding region, that's where we identified these three mutations in many patients. I think more than 50% percent of patients were just carrying this mutation. So interestingly, the functional study would tell us this. If there is just correlation between factor for the virulence of this disease for the affected patient. Uh, Actually, if you read the full paper, you will see that we try to explain each and every patient that we sequence. Actually, the three mutations we identified in this patient, that those who died, and also there are those who recovered. But maybe they recovered due to the fact that they got the antibody, monoclonal antibody, and the other antiviral treatment that I mentioned here. But we registered most of the death during the first week. And I remember during that first week, the case management, we did not have yet the possibility to give some treatment option to the patient. But again, on the molecular level, I can't give you enough information without just functional studies to see if there is maybe inhibition of function of the genes, if the gene expression is being affected. But interesting finding just here shows that these genes were located in a really close to coding region, which could be a very next target for further studies, for functional studies.
Thank you very much, Prof.
No,
Maybe
this was a
maybe
very good question. Yeah, yeah. Go
yeah.
ahead, go ahead, go ahead,
Yeah,
doctor.
can I also ask you that is
Of
there any
course.
correlation between the
Mhm,
mutation that you have seen, the 3 synonymous mutation that you have seen, with the
mhm.
virulence of the virus?
We don't know. We don't know yet. That's what I was mentioning. We need further functional studies to
Okay,
tell us. But
okay,
surprisingly, majority of these patients, they are carrying the mutation, the three mutations. That means maybe these mutations are putative because they are putative in the region, which could be, as I mentioned, they are not located in the coding region. They are later located in non-coding regions, in the splicing site. They could be just suppressing maybe the protein translation or gene expression. So we don't know yet.
Well, thank you very much, Prof, for that
Yeah.
clarification. So I'm
Yeah.
still checking
Pleasure.
through if there are other questions. I can't see anyone at the moment.
All right. It's a
So thank you
pleasure.
very
So
much,
you can
Professor.
just share. Thank you very much for inviting me to talk to share the experience from this recent outbreak. It was really a pleasure to share this talk with the audience here. I appreciate it.
Yeah, thank you very much, sir. So
Thank you
before
very
you go,
much.
sir, let me, of course, let me just appreciate you for giving us that, wonderful presentation. And like I said earlier, we have gained a lot. You gave us a lot to process at the same time, and also let me also say my big appreciation, or my big appreciation goes to the organizers, the backroom staff. I can see that your effort is actually leading to the resounding success that this webinar is recording. Thank you very much. And also to the audience who has been with us from the beginning. Thank you very much. And those people that ask questions, thank you very much for asking your question. And I hope that we'll see you again in the next episode of Install Africa webinar. Thank you very much. I don't know if the MD has something to add before we call it a year, because now.
Network is very bad. I'm not sure if you can hear me. First of
Very
all,
well.
I
We
will
can hear you.
appreciate. Okay, I appreciate our Professor Leon Mutesa, for this wonderful presentation. We are very glad that you welcome our call and you were able to present excellently. Thank you very much for your contribution. We are glad with
Pleasure.
all your support that you have been giving us since the beginning of this program, this project, and we hope
Okay.
that we will still be coming, and we know that you are going to continue to support us. Thank you very much. We are very grateful. And I will also thank our
My pleasure.
audience on the back end and those watching this webinar currently on the YouTube channel. Thank you very much for your support. Please, this is the ending. This is ending for this year. So we meet again next year. Thank you for coming. We appreciate you all. And watch out for our Firat Biostatistic Research and Methodology Clinic. And this will be played immediately after this program, and for you to have an insight of what we are bringing for you next. Thank you very much for all your support. My colleagues that are here, thank you for your support, for your contribution. We are glad to have you all. Thank you.
Thank you. Okay. See you
Thank
next
you.
time. Bye bye.
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 Bye bye.
Thank you so much. Bye bye. Thank you.